All right, I think we can get started. Thank you for joining us today. I am Heather Gatney, and I am the coordinator of the CVFS webinar series. Our host today is Coulter Mitchell, Associate Research Professor at the Survey Research Center at ISR. Coulter is going to give an overview of the design of the genetic data in the CVFS. The genetic data were collected when the Nepal-specific World Mental Health Survey, the CD, was administered. And if you missed it, we had a webinar on the CD back in September that you can watch now on the CVFS website. Also, very quickly, before we get started, I would just like to give everyone a heads up that 2 weeks from today, on December 15th, we will be having our last webinar of the year. We'll start back up again in January. But in 2 weeks, I hope that everyone can join us. It's going to be a little bit of a different format. It's going to be more of an open discussion where we talk about future proposal and research ideas. And we're going to have 3 different presenters next week. Okay, so for today, we will have a Q&A session after Coulter's presentation. We ask that you remain muted until then. During the Q&A, you may unmute and ask your question, or you can submit a question anytime today using the chat feature. We'd also like to remind you that this webinar is being recorded. If you have any objections, you can disconnect at this point. And then a recording of this webinar and the slides will soon be available up on the CVFS website. And so with that, I will hand the discussion over to Coulter.
All right, thank you, Heather. Everyone can hear me okay? Yep. All right, so I'm going to ask that maybe we have a slight modification of the typical rules. If you have an issue coming up, maybe you can raise your hand, or I may not be able to see the text as if you send a question earlier on, in part because I might stop a couple times through here just to make sure everyone's where they need to be, because we're going to be covering things that I'm assuming most people don't have a lot of experience with. So please feel free to make this maybe slightly more interactive than we normally would, if that's okay. And Heather, feel free to stop me if there's a whole bunch of questions and I'm just missing them. So yeah, we're going to be talking about genetic data in the Chitwan Valley Family Study. I'm really excited. I could go on and on about a long story about a young graduate student who thought this was a great idea, and had people say, well, come back in a little while, maybe we could think more about it. Yeah, that was targeted at Bill. But
no, you're evading me to like interrupt your flow, and I can only conclude, Coulter, that you don't actually have 40 minutes of content, and you want me to say 20 years was the right amount of time to wait. Yeah. And it worked really well.
Yeah, it did. It worked great. Okay, so with that start, I'm going to give a little bit of content on motivation for why social scientists, health scientists might be interested in genomics, genetics. I'll give a very basic genetics refresher, but it's actually really important to have a basic understanding of what this looks like, because it's probably not what you think it is. We'll talk a little bit about this Chitwan Valley Family Study specific design, and then we'll show some applications, some potential applications when the data become available. Okay, so with that, there's been a lot of work actually about looking at biological consequences or biological correlates of social disadvantage or health. And this is, 15 years, even 20 years now, a lot of biosocial work. Initially, this work kind of focused on larger systems like blood pressure, cholesterol, glucose level, but it's rapidly moved to more molecular level and brain. And the molecular level is what we're focusing on here, genetics. One thing to keep in mind, and one thing that makes CVFS so exciting, is that not all populations are equally represented in biological work. So why would social scientists care about biology? A lot of times we talk about it in terms of mechanisms. Genetics doesn't really fit in totally here because genes are constant across our lifetime. But it still could help tease out some mechanisms, and we'll talk a little bit more about that later on. But that's often one reason that people are interested in biology more broadly. Or indicators, can we have some sort of predictor of a future outcome? long before we actually see that outcome. I mean, that's kind of a dream of having some biological indicators. Genetics could be considered some of those. Or even just controls. Can we have better measurement of the thing that we care about, say some social influence on a behavior, by adjusting out some of the genetic confounders? We'll talk a lot more about moderation. We see a lot of heterogeneity of response to different contextual effects, for example, and how does that then, how do we explain all that heterogeneity? And sometimes that's biology, or in this case, genetics. And then there's also this other component that a lot of people are interested in, which is, again, not all populations are equally represented in medical research and genetic research and biological research in general. And in order to improve our understanding of biology and medicine, we need to have more generalizable results. To give you a sense of what I'm talking about, whose biological data we have? This is actually a great paper by Alicia Martin, who is actually working on the CVFS data at Harvard, getting it ready. Anyway, in 2019, she led a study that looked at how many people are participating in genomic research. And this is Genomewide Research, which you'll hear a little bit more about later on. How many people is kind of that top graph over time, and the bottom is what proportion, because it's double counting some people. So what proportion of people who are participating in these genetic work are from different ancestry groups. The column on the right is actually the global proportion of that population. And then you can see by just looking at, for example, European is that red box, you can see that the vast majority of people participating in genetic research as a proportion and as a count are from European populations, much smaller from every other population, with East Asian being slightly larger. And so that has consequences for our knowledge base. To be fair to genetics, that's true for brain science. It's true for other genomic sciences. It's actually true for psychology and a lot of medical science as well. These are other citations that talk about that. So in thinking about that, keeping that all in mind, and why this is potentially really exciting to have data like this in Nepal, we have to take a step back and talk a little bit more about genetics in general. So I'm going to start at the very, very basic, but we'll move quickly. So the simplest form of genetic variation that we have is called a single nucleotide polymorphism, or a SNP. So you might hear me say that. That's this classic CGAT, if you remember that, or remember from Jurassic Park when they covered that there. There are other forms of genetic variation, but this is the simplest form. A SNP occurs about 1 in every 300 base pairs. And that's what that C and G are, that's a base pair. And that's because most of our base pairs are actually identical across most sites for all humans. And then we say in 1% of the population, that counts as being a polymorphism. Anything less than that, it's too rare. One reason for that is that this is just a graph that talks about where we expect to see genetic effects. Most of the time, we think of, when we talk about genetics, we're talking about something like a rare allele, that someone has a rare genetic mutation and that causes a certain disease. Those are actually incredibly rare genetic mutations and have very strong effects. So that's that upper left-hand corner. The reality is most of our genetic variation is on the other end, what we call common variants. So more than 1% or more than 5% found in the population. And those have incredibly tiny effects individually. So keep that in mind, that actually most of the time we're talking about very small effect sizes for more common variants. So coming back to the single nucleotide polymorphism, we have 2 flavors of these SNPs. They're called alleles. One is the major, one is the minor. So the C or the G, the reason why they're determined to be major or minor is usually what population they're more common in. So which one is more common is usually the major, the minor is the less common one. Another piece of information to keep in mind is we inherit 2 pieces of information, one from each parent. So when all is said and done at a single SNP, we actually have either 0, 1, or 2 of the reference alleles, or the major or minor alleles. So that's the way we typically think about it. A single genetic measure, and it's either 0, 1, or 2. So it's a pretty simple measure. But what we do is actually a little bit different. Instead of genotyping that's more time intensive like we used to do not that long ago, we actually use far more automated systems now. And we can assay, using the technology that I'm showing down there, hundreds of thousands to millions of Snips. And then we can actually use a different strategy that I won't go spend a lot of time on, of imputation, to up to 30 million Snips. So keep that in mind. We're talking about millions of these variables. And that's really what SNP data looks like. You have SNP 1 through SNP a million or 20 million by a matrix of all of the people. In fact, we actually transpose it usually. So SNPs are the rows. So that's what a SNP data set looks like. It's enormous, and most of you will probably not be looking at it, but that's what we have. And actually, we currently have everything genotyped for Chitwan Valley Family Study, and we'll talk a little bit more about that right now, or shortly. So how does a genome-wide analysis typically work? That's really what the basis of genetic work today that's done. And what that is we start with what we call a discovery sample, and it's a very simple regression analysis. Imagine those, we'll say 1 million SNPs, and we have some outcome that we're interested in. We have a few, very, very few controls, and we do quite literally 1 million regressions of taking that genetic variation and predicting the phenotype, the outcome of interest. We then adjust for the fact that we've tested things 1 million times, and we have that's called a genome-wide significant. That's a p-value of 5 times 10 to the negative 8. So a very, very small p-value. And that's called a significant or genome-wide significant hit. We look at some other clustering and correlated markers. A key thing here is this is often done in a meta-analysis of not just one study. We wouldn't do a GWAS in CVFS alone. You would combine it with potentially dozens or even hundreds of other studies. And we'll talk a little bit more about that, but that's a meta-analysis of potentially hundreds of thousands to millions of people as well. And then there's a replication attempt in a separate external set of studies, smaller sample size, and that's also often so a meta-analysis. And the way this is visualized, I won't spend a lot of time on this, but the way GWAS is visualized is you have a chromosome on the x-axis here. This is like the physical location on your genome of where something happens. And this is that negative 10 negative log p-value. So the higher it is, the smaller the p-value. Each dot is a SNP, like we were talking about before, and it's that p-value, the p-value associated with that SNP and the phenotype of interest. And the height is how strong that association is. So keep this in mind when you're imagining, we're talking about GWAS. What does that mean? This is what they're talking about. Very exploratory and data-driven. So I have a lot of slides in here that for people who want to go back and look at this in a little bit more detail, and that's fine. Some of the strengths and limitations of meta-analyses, which is very common for GWAS, that I think a social scientist and health scientists, population scientists get nervous about are, it does significantly boost power and reduces false positives. It's great because due to imputation, we often have the same exact measures of SNPs. And actually we can share results really easily because we don't have to share the raw data, we can just share the results. Some of the limitations you might very quickly pick up on is not all cohorts measure things the exact same way. So we have to end up having very rough phenotypes. It does assume that there's a common genetic effect and that it's not different by, say, SES or by sex or other factors. So you don't have any interactions like that. And it limits the complexity of analyses, longitudinal analyses, for example, even adjusting for different cohorts or covariates. One final note that's really important to keep in mind here is you're going to hear me say things like ancestry. And that really shouldn't be confused with race, ethnicity, or in the case of our study, caste. Race, ethnicity, caste are socially constructed and are often the measures that we use when we look at social and health inequalities. It's not that they're not real in their effects, they're just socially constructed. That is sometimes correlated, sometimes not with ancestry. Ancestry is variation in genetic architecture, so frequencies in those SNPs between populations. That's what's primarily used in genetic data. And that results from many generations of demographic processes, which you'll note are under social control to some degree, including migration, fertility, and mortality. It should also be clear that genetic variation does not necessarily mean that there's a causal effect of biology. You can have small variations in alleles that are totally unrelated to an outcome, but will appear to be related. So that's one of the major issues that we have in genetics research. And the number and homogeneity of ancestors is very different by race and ethnicity. So what does this look like? In genetics, this is called population stratification. For those of you who are used to doing principal component analyses, this is a really nice example. This is raw genetic data, like genome-wide data rather, from people who have lived in European countries for 3 generations. And they just took the first 2 principal components of the genetic data, nothing fancy. And then you can see if it gets transposed and you color code it by country, that it very quickly looks literally like the physical map of Europe. And that's because people who live closer to each other, who have been around each other for many generations, are going to look more like each other in terms of their genetics. So it's actually a really strong predictor of that kind of relationship, geographic and historical. However, in other populations, like in the United States, it should also be known that like they're not homogeneous populations, right? So people can self-report as, let me give you an example over here of white. And if you look at this color coordination, these are different genetic ancestries. What I can tell you is that for people who self-report in white in a study in the United States, they will predominantly be from either, or their ancestors will be from predominantly Northern European or Southern European families or histories or genetic architecture rather. However, and so that's pretty homogeneous. If you go to someone who self-reports as Black, there's a mixture of European and African, and even in this case, a couple different groups in Africa. So it's far more complex. If someone self-reports as Hispanic, there's even more complexity, a mixture of a lot of different ancestries. So that's just to be clear, ancestry and race or caste in this case may not be exactly the same. And this is going to be a really fascinating study to kind of look at these and how they're correlated because of the migration and historical family relationships that you see in Chitwan. Okay, so I know I've taken a lot of time just to get to, let's talk about what CVFS has actually collected. And as Heather already mentioned, I think the most important thing for me to tell you is the genetic data are a subset of the CD mental health data that we heard about on September 29th. So I'm really not going to spend a lot of time talking about it because I think there's a great example of how that was collected and all of the questions that are related to it. So the survey design, the study design should really be focused by going back to that. And then I'll also add that if you're thinking, oh, but I want to do the genetic data and use it with something else in the Chitwan Valley Family Study, luckily, we also had a different presentation on how you might merge across different parts of the study. So that was also presented on October 13th. So we have those, please go back to those, look those up. So this Chitwan Valley Family Study genetic data was collected during the CD survey, as we were saying before. We collected saliva. And one thing to know about that is the genetic data is the same in all of the tissues, unless it's a tumor tissue. And so it's really whatever is the easiest to collect. Saliva is incredibly easy to collect, and it's very robust. It can be stored for months at room temperature with very little problem. And so it's a really good solution for collecting when you can't put something immediately into a freezer, for example. 96% of the CD participants consented to providing saliva. This is far and away higher than I think any study I know of. Normally, at least in the United States, it's closer to 80 to 85% of respondents participate in saliva. So this is very, very high. An important thing to keep in mind if you're interested in this, the ethics around collecting genetic data have received a lot of attention. And so as part of kind of standard NIH funding and IRB requirements, collecting genetic data has its own consent. That's very different from the study participation. So those are two separate things. And then there's actually even another consent related to being willing to share your genetic data and kind of in repositories, de-identified data, of course. And so it's a lot more complicated than maybe just thinking it's just an add-on to a survey. There's a lot more thought put into it than that. Just to tie back to the CD, this actually comes from a paper using the CD data. I just wanted to provide this as an example of, this is the distribution of the CD data. We would expect, since 96% of people participated in the saliva, that these would be similar percentages for people who participated in genetic data. We don't have that finalized yet, but that's what we're assuming. It would look something like that. So there's a paper you can go back to try to get a sense of how many of different groups you might have. So the DNA, after it was collected, it was extracted in Nepal. So you spit into the saliva tube, as I was mentioning before, in this case, an origin tube. It sits around for a little while, and then it gets moved to where we extract the DNA out of the saliva. It's a much smaller vial. It was then shipped to the Broad Institute for genotyping, as I had shown you before what that looks like. And that's actually been completed as far as I know. I think it's all been completed and is now working. It's with Jordan Smoller, who's the PI with Bill with that grant that collected the data, and it's now with them. They are what we would call cleaning the data, going through quality control, and that's in concert with the Psychiatric Genetics Consortium that also is helping to kind of link us together with other psychiatric genomics studies so that we can participate in these GWASs. Bill, did you want to add something to that?
Yeah, they had to be frozen at minus 70 degrees or something like that, Coulter.
Yep.
And air shipped from Kathmandu to Boston at this minus 70. Quite an undertaking. Dirga and certainly the team at ISCRN deserves a lot of credit for all the mechanics involved in making this physically possible. Broad Institute receives genetic samples from all over the world, and they do a QC on arrival. They went through every sample and determined the Chitwan Valley Family Study quality of arriving saliva samples was as high as they have from anywhere. And they also did a bunch of QC in the extraction process and report that went very, very well as well. And so Coulter, 100% accurate, but as usual, I just wanted to embellish a little bit.
No, and you should. Yeah, no, I'm excited. It's a key aspect of this, that collecting genetic data is becoming very common, but the quality of collection is actually a lot harder to pull off, and by all counts.
Thank you for including your graph that shows it's common in European ancestry locations, not super common in South Asia, and no small trick to get it from Chitwan to the labs in Boston. And we're very, very happy with how all that went.
Yeah, no, it's actually quite. Here's what I'll say is, like, we'll be going to some applications shortly, and that's what we'll spend the bulk of the time talking about. But if this were just a study of 5,000 even unrelated individuals in Nepal, that in and of itself would be a pretty amazing study because it's so unique. It's a lot more than that. And that's about what we'll talk about next. So one of the really cool things, as you know from CVFS, is that there's a lot of family relationships, and that's really interesting and has a lot of opportunities for genetic research. So these are what we kind of expect in terms of our sample sizes. We're still finalizing this because we haven't finished all the QC, so these numbers will definitely change. But this is some rough estimates for what we expect in terms of what we call trios. which are mother, father, child. These are actually the number of individuals in trios. So there are actually more trios in this because if a mother, father has multiple children participating, that's actually multiple trios, but this is just counting individuals. And you can start going through this like one parent, but greater than one SIB pair, there's 1,435 people, parent-child duos, 618, and all the way down to unrelated, it was a little bit less than 2,000 out of the total of 10,308. However, in talking with Dirga and a few other things we've been trying to estimate, we think that there's probably closer to like 2,000 trios, which is an enormous study for trios. We have probably somewhere north of 5,100 mother-child pairs, over 4,500 father-child pairs, and probably a lot more than, but probably at least 2,000 sibling pairs, which again, all of these are some of the biggest that I've seen in these kind of family relatedness studies. So there's some really cool things to keep in mind. Okay, so let's talk about some applications that might be of interest for you. So now at this point, I want you to kind of sit back, think about what you care about Chitwan Valley Family Study, what you're interested in studying, and then I will then come in and say, okay, here are some applications for the genetic data that you might consider find interesting. So if you remember correctly, I started to talk to you about Snips, right? And they have very tiny genetic effects, so tiny that they would explain almost nothing in terms of the variance, even if they're highly significant. So what do? So what we do is we take these external GWASs, right, that's from another set of studies that we didn't participate in, say of height or type 2 diabetes or cardiovascular disease, any kind of outcome that you want. We take that GWAS and we actually sum together a weighted version of the score for the entire genome. And so each person gets a score that's essentially a predictive score of the probability of, say, being taller or having a certain disease or something like that. And so what we do is we create this polygenic score. So it's a single index of an entire predicted probability, essentially, of having a certain disease. What does that look like? That takes that outside GWAS. Remember the Manhattan plot? It takes our data, and instead of saying, okay, here's our one SNP, which would have a small effect, you add up multiple SNPs and you have kind of more of a normal distribution of SNPs, a much more predictive score. There's a lot. There's literally, you could spend weeks, months, years focused on how to create these scores. Don't worry about it. Other people will take care of it. But if you want to dive in more, there are papers on it. One thing to keep in mind, though, is we have generally found that polygenic scores, while sometimes very predictive within homogeneous populations, are not portable by ancestry. An example of this is up in the upper left-hand corner. This, again, is a great paper by Alicia Martin. So height, at least in the United States, is not very different by, say, African and European ancestries. We don't see any phenotypic difference. When we look at the polygenic score for height, it looks like European ancestries would be between 4 and 6 inches taller than African ancestry. Why is that? Well, there's a lot to be talked about in there. This paper is a great example, but the short version of this is it's not about the underlying biology. It's more about what we call algorithm bias, that the people who are participating in the studies have lots of other things that are correlated with height and spuriously with genetics, and they end up embedding this algorithm bias, this selection bias into the algorithm, and that produces an algorithm bias. So it's a very common thing, but by having more diverse samples like Chitwan participating, that improves the portability. But for right now, don't be thinking you can use a genetic score to then say, oh, this is why this cast is different from this cast. That won't work. Just don't try to do that right now, because we're nowhere close to being able to do that. So keep that in mind. This is a lot of what personalized and precision medicine is based on, which maybe should scare you a little bit, because it's not as predictive in some populations as it is in others. keep in mind that the typical trade-off is the higher the prediction, the less the biological insight. In other words, like because you're kind of compressing all this biological information across the entire genome, it may be more predictive, but you've kind of lost like what aspects of the disease are operating. But again, we can spend a lot of time talking about this, and just about every phenotype you can ever imagine has already been GWAS'd, sometimes well, sometimes not. But that's what we could be thinking about. Lots of reviews for you to come back and look at and commentaries on polygenic scores. You might also be interested in gene-environment interaction and gene-environment correlation. The G by E is gene-environment interaction, and gene-environment correlation is denoted by RGE. So again, a lot of reviews that I'm just going to put up there if you want to go back and look at them, because it's been a big topic on how to do gene-environment interactions. I'm only going to say one thing, and this is actually purely from a statistical point of view. A lot of times, and this isn't just genetics, but we misunderstand how to think about interactions. A lot of times we get these intuitions from experiments that don't carry over into the field. And one way to think about this is that we don't really push people to the extremes and have 4 equal-sized extreme groups to be able to test, say, an interaction. And there's a great old study, 1993 in Psych Bulletin about this. But the rule of thumb on this is that if you are doing an interaction and your interaction is the same size as your main effect, you need 4 times the sample size to detect that interaction. That's rare to have that. If the interaction is half of the main effect, which again is a pretty good size interaction, you need 16 times the sample. So it's very large sample size requirements. And remember, these polygenic scores and sometimes the environment don't have sometimes very large main effects to begin with. So keeping that in mind, gene environment interactions are going to be challenging, but this study still does have a very large sample. So it is possible still, but be careful. Maybe more interesting and something to consider are gene environment correlations. That's that RGE. That's actually tremendously important and tremendously interesting. You can kind of divide this into 3 different groups. There's active, and that's where genes are choosing, a person's genes are driving them to certain environments. Evocative, where the genes are manifesting in some way that then the environment is responding to something, right? So oftentimes the example people give is, you have genes that are related to a child that's more fussy, and then the parents respond to that. I don't know. Anyway, there's different ways of thinking about evocative. And passive we'll spend a lot more time on, and that's where genes from the parents are passed down to the child, but so is the environment. So they're kind of passed together. And that one ends up becoming a really cool one to think about with families. So one example of this, there's lots of them, but this is a nice one using a study from Dunedin, New Zealand, and they used genetics of education and ended up showing that people who had different genetic scores were more likely to migrate to other countries for education, and that was true even in different environments. So there are lots of examples of this, but think about how it's genes maybe actively selecting in some ways. But that's a gene environment correlation, not an interaction. If you do decide, I really want to go down the gene environment interaction route, what I would recommend is think on a more macro level, bigger things, which Nepal has measured really well. CVFS has measured really well. Here are some examples of, say, using the Vietnam era conscription, something more macro like that, or historical changes like, well, I don't know, armed conflicts and how that might interact with something. So those big macro level environments are exogenous, and you have a little bit more heft to be able to talk about your gene environment interaction. Oh, whoops, that was supposed to be up above. So this are the family models. Heather, I'm going to send you a slightly modified version of my slides. So these are those family models we were talking about before. And just to remind you of the set of different family types that we have. One thing that may not be of interest to most of you, but could be, is that by having a trio and we can do analyses controlling for the mother and the father, we can actually break the effect of the population stratification and be able to compare across populations. So there's some really nice kind of genetic things we can do that way. It's not a, you know, it's only, we might only be a sample of 2,000, but there's some really powerful things to be able to examine that way. So that's why we like trios from kind of an exploratory analysis. But let me show you a couple other ways in which these family data will be really helpful for doing your work. One is what we call genetic nurturance or genetic nurture. And that is that these GWAS are clearly, the GWAS of these different outcomes that we're interested in are clearly influenced by the social environment. So they probably pick up more than just biological influences. So how do we look at that? There's an example, a couple different examples. But this first paper by August Kong down here, The Nature of Nurture, the Effect of Parental Genotypes. The point here was to look at, we know for sure that the genetics that a parent has influence the genetics the kid has and then influences their behavior. That makes sense. But what about the genetics that are part of the parent that they don't give to the kid? Do they still influence the environment in which the child is raised? And that's what this nature of nurture is looking at kind of the untransmitted alleles, so to speak. So let me show you an example of one that was published a couple of years ago. So this is like passive gene environment correlation, genetic nurturance. This looked at genetics around education and how that was related. That's the upper box right here, is related to the child. And that's correlated at 0.5, which is what you would expect. And clearly that had then effect on how far this person went in school. That's all stuff that would make perfect sense. What was interesting was that even after controlling for that, there was still a direct effect of the mother's genetic data on the child's educational outcomes. And how did that work? Well, it was mostly mediated by the amount of cognitive stimulation the mother had in the home, in this case, in this particular study. So you can start to imagine, is there something about the parents' biological makeup, their genetic makeup, that maybe is also influencing their environment that they're raising their child in addition to contributing genetics. So it's kind of a cool way of testing this. Another way that actually is quite powerful for looking at genetic differences is common within the social sciences already, which is sibling comparisons. So sibling comparisons, and these are not monozygotic twins, in other words, identical twins. These are either fraternal twins, dizygotic twins, or just regular siblings. Controls for any influences shared by the siblings growing up in the same household. But it also is kind of a cool thing that any slight variations in genetic differences between two siblings is random. And so there you have this nice causal estimate of differences in genetics. This again is a different study. And I just gave an example. They have the darker bars are the main effect of the genetic polygenic score on education, occupation, and wealth. But then the sibling differences in those, and those are the light blue bars in different studies, eRisk, AdHealth, WLS, that all have siblings. And you can see it attenuates that effect, but that is a more causal effect of that genetic variant or that polygenic score. Okay, so I'm actually wrapping up right now. I feel like I went through a little too fast, maybe. Covered a lot here, and I'll love to talk more about what you're interested in, but just to be aware, if you're really interested in like doing social science and genetics, here are a couple groups that you might want to look into. There's an NIA biomarker network that does a lot of work with genetics and other biomarkers. Social Science Genetic Association Consortium, which is exactly what they describe. Psychiatric Genetics Consortium. If you also want to learn more, I'll just put a plug in that I help run a course every summer, 2 of them, on genomics for social scientists. And that's both genetics and introduction to genetics. That's a week long. And epigenetics, which is 4 days long. And there's the website to go look at that. And with that, I think I'm done. So I'm going to stop sharing unless people want to see more slides. We can go back to those.
Thanks, Coulter. Super helpful, and thanks for covering so much of that territory while while I give others a moment or two to consider what questions are on their mind. Could I ask you one? When I first met Jordan Smoller, our collaborator from psychiatric genetics, he was giving a presentation to the World Mental Health Consortium, and he made a statement that I should have checked with you to validate this, but I'm curious. He opened that presentation with a statement that was inheritance of mental disorders is higher than inheritance of breast cancer, which got everybody's attention. Now, my little I know is that actually inheritance of mental disorders, as best we can tell, varies quite a bit across mental disorders. But, thank you for giving us a presentation about the general potential of genetic data to contribute to health research and population research. What do we know about the kinds of things that are more inherited and potentially genetic than other things? Do we know much about that?
Yeah, no, that's great. We could spend a lot of time talking about the different substantive like differences, but you're right. So one thing to keep in mind is when people say something is heritable or not, an interesting thing is that we often interpret that as being like an individual level variable, like it's inherited because it feels like it should be, but it's actually not. It's a population level estimate, which is why it can differ over time. It can change over time. It can also change by population, and dramatically so, actually. The other thing to keep in mind is most of the time when people talk about heritability, they're talking about using twin studies to estimate that. And a lot of times there's actually this whole body of research that talks about missing heritability, because what we thought was that we had high levels of heritability using twin studies, high levels of heritability of a lot of different things, including mental health is quite high. He's right, higher than breast cancer. That's totally true using twin studies. But then when we started doing molecular genetic things, we were nowhere close to having that much variance explained. And in fact, currently we can explain more genetic variance with molecular measures than we for breast cancer than we can for psychiatric outcomes. But maybe not schizophrenia. I think that might be close, but
that was my understanding. Schizophrenia is like one of the most I'm using words I don't
heritable.
I understand. Heritable.
It's both heritable and also has some of the biggest genetic effects, right? But even still, and this is one thing to be careful of when we're talking about genetics. Just because something is heritable and your parent had it doesn't necessarily mean that the genetics are going to work out that way for you, because again, it's a population level parameter. And so that's really important to keep in mind, because a lot of times we're talking about alleles and we know it's directly passed on, but that doesn't necessarily mean that the phenotype is passed on. So yeah, Bill, this kind of goes back to that discussion of prediction versus biological insight. The larger our GWASs are, typically the more predictive our polygenic scores have become. And so in the United States, we can use in European ancestries in the United States and in Europe, we can use a polygenic score from educational attainment and explain 20% of the variance in cognitive outcomes in children. That's pretty high, right? And being able to explain 20% of the variance. But then you go to something like depression, and in African ancestries or Asian ancestries, and it's probably 3 or 4%, right? So it's that much variability. And so a lot of times I feel like we're still sorting this out. But the more studies we have, the more we've found that we can explain. But psychiatric sorts of outcomes have been some of the harder ones for us to do for some reason. Cardiovascular study and anthropometric, we've gotten better at those. But again, mostly in European ancestry, right? African ancestry might be a third of the prediction.
Well, and I'm pretty simple-minded on the survey side of trying to measure these things. And Emily might like to complain about all the measurement error in getting children's heights. But I bet we do a lot better at that than we do at measuring mental disorders, based on what I know about the science of measuring mental disorders.
Right. Yeah.
I'm going to give other folks a minute or two to consider a question before I come back with the next one.
That's all right. I do worry a little bit. I just turned a fire hose on, but
Coulter, I'll describe that as a digestible fire hose. So
okay.
I feel like I need to think through everything that you said before I really have any good follow-up questions. But thank you.
Well, so I'd be curious if people do have questions. I mean, I think one thing that I would recommend is if you do have something that you're interested in, some outcome that you're particularly driven by, let me know. I can point you in places where you can look up whether or not there's ever been a GWAS, and you can look and see who was in that. You can read the study, see how it was measured. One thing to note is that in addition to the ancestry sorts of things, most genetic research is conducted on adults for obvious reasons. So normally what we would do is you would say, oh, here's something related to type 2 diabetes in adults. What is it related to in child development or in adolescent development or early adulthood? And that's what's kind of exciting, is teasing out maybe the disease process.
I'll say I feel you were quite correct to explain the complex consent process that's separate for genetics. That in our study we got consent for the survey. We did the survey. We have 10,714 complete interviews. Coulter showed you 10,308 complete saliva collections, and that difference are people who didn't give consent for the saliva collection. But we did include parental consent and did get parental consent to get a whole bunch of 15 to 18-year-olds, which isn't everything, but it's something to be able to study some of those trajectories.
Yeah.
Coulter, I guess my other question is about the science of polygenic risk scores, and maybe I'm spending too much time around statisticians, but my understanding is that's grown to be more controversial over time. You kind of pointed to one of the reasons for that in what you showed, that so much of the data comes from European ancestry. Does it do well in Asian ancestries? I'm very glad to be working with the PGC, the Psychiatric Genomic Consortium, because they are engaged in a global scale. They keep trotting out polygenic risk scores, or PRS, with little subscripts, PRS-CX, if I may. And I would love a 2-minute version of I have an understanding, but I would love to validate that understanding of why that's better than a PRS in this situation.
Well, so the CX one I'm thinking is the cross disorder one. Is that right?
I think there's a cross ethnicity one.
And there's a cross ethnicity one. Yeah. Okay.
I might have the wrong subset, but I think if I understand right, in addition to the European ancestry archives of psychiatric genomic data, there are some now from Taiwan, Japan, other places in Asia that they're now using to construct these cross.
No, that's right. That's right. So the thing to keep in mind is genetic analyses within a homogeneous population. That's the bread and butter of what this is. It's just that essentially for a longest time, for reasons that we can't spend too much time on, it was all European. That was the way that they handled it. European ancestry is very homogeneous compared to almost every other ancestry, so easy for genetic purposes. But what we're finding is that when you do these kind of separate GWASs in these different homogeneous populations, and then you meta-analyze them, you actually end up creating a new GWAS that's more generalizable to the entire human population. And then you create better polygenic scores because of that. So that's what's happening. So it's essentially like break everybody out into their homogeneous ancestries, analyze them, meta-analyze them. And then so you can imagine a situation where, like in the United States, or for example, in Nepal, where we might have 4 or 5 different groups that are more homogeneous in terms of ancestry. We analyze them separately, then we meta-analyze them, and then those are the Nepal results. So that's the way it might have to happen, and that does seem to work better. And
Yeah, I'm placing my trust in the experts in that science, but yeah, it's an interesting concept to try and get those to work better.
And part of it really is, there's some great papers, I should have included them, and I just forgot her name, it slipped out of my head. Anyway, just showing that it's not really about the genetic architecture. It really is about who is participating. It's about selection bias on who's participating in the different studies. An analogy I often use is, imagine that you were building a voice to text app, but you only had people from Boston creating that app, right? And that's what it was training on. If you then bring it to Michigan or to my home state of New Mexico, it's not that we speak a different type of English, or that it's bad. I mean, we can get in arguments, but whatever.
I'm not going to distract you with that.
Yeah. It's just that it's not the same as it. So it's like it wouldn't work quite as well. And that's essentially what's happened, is we have this kind of selection bias that leads to an algorithm bias. And so by being better about being more inclusive and more generalizable about who's participating, we're finding that we have less algorithm bias on the output. But it's really hard.
I'll try to feel good about contributing to that, no matter how we do on our own science, by putting these data into the genomics consortia, plural, so they could be used as widely as possible.
Yep. Yeah, it'll be great.
Well, a couple people have agreed with the overwhelming nature of the information flow, Coulter. I will say, in 2 weeks, when we return to kind of brainstorm about future research, I fully intend to open with this one and bring us right back to this spot, because as you showed, the triad, dyad family composition of our genetic data opens a lot of great new doors that I think are going to be of interest to many scientists who are involved.
Yeah, and I would say, even if your goal isn't to study the genetics of it, simply having it there as a control strengthens your argument that it's not due to genetics.
Yep.
I'll certainly discuss that some when we talk about the project we have right now on parental mental disorder predicting kids family outcomes.
Yeah.
About out of time. Thank you so much, Coulter.
Yeah.
Thank you, Coulter.
Thank you.
All right. Bye, everyone.